Well, hello and welcome to everyone joining us for this X-Talks webinar. Today's talk is entitled A New Paradigm in the Detection of Amyloid Pathology, the Emergence of Highly Accurate Blood Tests. My name is Corey Stanton and I'll be your X-Talks host for this event. Today's presentation runs for approximately 60 minutes and the webinar includes an interactive Q&A session. To make the most of this session, feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to as many as possible. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please don't hesitate to contact me by sending a message using this very same chat box. Please note that all audience members are in listen-only mode, and that this event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank C2N Diagnostics, who developed the content for this presentation. C2N Diagnostics specializes in diagnostics for brain health, offering high-resolution mass spectrometry-based biomarker services and products. These aid in clinical decision-making, improving patient care, enhancing clinical trial quality, and supporting healthcare research. C2N's assays have been pivotal in over 100 Alzheimer's disease and neurodegeneration studies globally, including landmark treatment trials. With ongoing collaborations with major pharmaceutical firms, C2N has contributed over 15,000 PROSIVITY-related biomarker measures to peer-reviewed publications with more manuscripts in review. And now I'd like to hand over our moderating duties to Sheena Arora, who's connected in over the phone, and she'll introduce you to our panelists. Sheena, the floor is yours.
Thank you very much, Corey. I'd like to take this opportunity to welcome everyone. And we have two great panelists, Dr. Rabinovich, who joins us from UCSF, where he directs the Alzheimer's Disease Research Center. He's played a pivotal role in PET imaging and making it available to patients through his seminal research. And the fun fact about him is you can see in the background there that he's a Cubs fan. So very happy to have him here. Next slide, please. We also have Dr. Bronstein, who is CEO of C2N. And Dr. Bronstein is a cardiologist by background, and he can tell you everything and anything that you'd like to know about the analytes. He's been in the Alzheimer's disease biomarker area since 2007, and he's also an avid photographer. So if anyone's interested in the eclipse and missed it, Joel has some fantastic pictures. So with that, I'd like to turn it to Dr. Rabinovich for his presentation. Thank you everyone for joining today.
Thank you very much. It's really a pleasure to join you this morning, especially because the Cubs are rapidly dropping out of the playoff race. So this is a very welcome distraction. But in all seriousness, I am excited to share with you today some of our experience implementing Alzheimer's disease biomarkers in real-world memory care. So outside of the highly controlled research setting, by sharing some lessons that we learned during the IDEAS study. The sort of prototypical Alzheimer's disease biomarker is amyloid PET, and amyloid PET has played really a pivotal role in Alzheimer's disease clinical research and in clinical trial design. And we do have 3 FDA-approved radiotracers for clinical use in the United States. These are florbetapir, flutometamol, and florbetaben. And especially with the advent of new disease-specific therapies like anti-amyloid monoclonal antibodies, amyloid PET also can play an increasingly important role in the clinic. And just in the newly published Alzheimer's disease diagnostic criteria, amyloid PET is considered a core 1 biomarker sufficient for diagnosing Alzheimer's pathophysiology. It's incredibly important for determining treatment eligibility for amyloid targeting therapies, and also potentially for monitoring the effectiveness of the therapies by determining when people have reached amyloid clearance. And yet there's a major gap between how amyloid PET is used in research and clinical trials and its use in the clinic. So for example, the FDA has approved amyloid PET interpretation based solely on a binary visual read of scans as negative or positive. Yet in research, we use central reads by highly experienced raters. We use quantification using a scale called the Centiloid scale, which I'll talk about a little bit. This is a uniform scale that can be used to interpret the the amount of amyloid PET uptake. And nearly all the data that we have about the clinical applications of amyloid PET have come from these highly controlled research studies. And so in today's brief presentation, I'll talk about what we learned evaluating the performance of amyloid PET in IDEAS, which was a real-world large study of amyloid PET's clinical utility, looking at the quality of images in the real world, the reliability of community visual reads, and the feasibility and results of quantification. And so for those of you who aren't familiar, the Imaging Dementia Evidence for Amyloid Scanning, or IDEAS study, was a US-wide single-arm multi-site longitudinal study that evaluated the clinical usefulness of amyloid PET in Medicare beneficiaries who had mild cognitive impairment or dementia of uncertain cause. Alzheimer's disease was considered one of the possible etiologic diagnoses, but the clinicians were unsure if the patient had or did not have AD. We enrolled over 18,000 patients between February 2016 and January 2018, followed them for 12 months. They were recruited from 600 memory clinics and scanned at 350 PET facilities. All the clinical reads of the amyloid PET scans were done locally, so there were no central readers. I should say that IDEAS was done in partnership with the Centers for Medicare and Medicaid Services under a program they call Coverage with Evidence Development, where they cover certain diagnostic procedures like amyloid PET as part of research studies that are evaluating the clinical usefulness of the new technology. And really the study was useful to Medicare in determining whether amyloid PET should be covered for their beneficiaries. So just to very briefly summarize the high-level results of the study, we found that amyloid PET very often led to core changes in patient management in terms of medications patients were receiving or how they were counseled about safety and future planning. We found changes in one or more elements of patient management in over 60% of patients with MCI and dementia, which was more than double the pre-specified goal of at least 30% changes in management. We also found that amyloid PET changed the diagnosis in over a third of patients, and 25% the diagnosis changed from Alzheimer's disease prior to the PET scan to a non-Alzheimer's condition based on a negative scan, and in about 10.5% from a non-AD condition to Alzheimer's based on a positive scan. We found that amyloid PET had a more modest effect on medical outcomes. There was actually a 4.5% relative reduction in 12-month hospitalizations in beneficiaries who underwent amyloid PET compared to propensity-matched Medicare beneficiaries who did not undergo amyloid PET. This potentially is meaningful given the very high morbidity and cost of hospitalization, especially in patients with cognitive impairment, but it did fall short of our pre-specified goal of a 10% relative reduction. We found no impact on emergency department visits and a neutral overall effect on costs. Yet with these data, and especially with the new therapies being approved by the FDA in October of 2023, Medicare retired their national non-coverage decision of amyloid PET, leaving it to the discretion of the local Medicare contractor. And so far, at least in San Francisco, we've had very good luck with Medicare covering this now as a covered benefit for Medicare patients. Now, in going from research to real-world studies like IDEAS, it's important to understand some of the major differences that occur between how PET imaging is implemented in a research study or a clinical trial versus clinical practice. So in research, we have central qualification of scanners, making sure they're up to snuff. We identify the image resolution of the scanner. We have tightly controlled acquisition time windows. So how long after injection of the radio tracer do you start the PET imaging? How long do you continue? How do you reconstruct the images? Often studies have structural MRIs that are used to pre-process the PET. And there are importantly central readers that are involved in this PET interpretation. Now, in the real world, none of this happens. So in IDEAS, we didn't pre-approve any scanners. We didn't actually know the resolution of the scanners that we were using. As I said, we had over 350 imaging sites. There were really variable acquisition time windows, reconstructions, no MRI for processing the images. And importantly, as I mentioned earlier, the images were read by local readers who had undergone some online training, but had, I would say, variable experience interpreting amyloid PET scans. And so here are some real-world amyloid PET data. We found some interesting scans that probably wouldn't have made it into a clinical trial. So here you see some interesting anatomic variants in the brain, major brain lesions. This is a stroke in this individual, so no radiotracer perfusing here. Very severe movement artifact that compromises the quality of the image. You can see errors in image reconstruction, some artifacts, corruption of the image. Occasionally, we had a cut field of view looking at our target region, which is looking for uptake in the neocortex, or the reference region that's used to standardize amyloid PET quantification, which is in the cerebellum, just large chunks of these brain areas missing from the scans themselves. Occasionally, patients were imaged with a wrong radiotracer. So this is an FDG PET scan that was uploaded as an amyloid PET scan. This is a phantom image, so it's not actually a human brain. And occasionally people image the wrong organ. So this is a full body PET. The brain is just a little teeny tiny element here, and it's actually FDG, not amyloid. So, welcome to the real world. It's quite different from the clinical trial world. That said, as I'll show you, we were actually very successful in the majority of cases in being able to use these images and generate quantitative values for the PET uptake. And this is a little bit in the weeds for those of you who are into neuroimaging, but because we didn't have an MRI, we had to develop an in-house PET-only processing stream to process and quantify the PET images. This was called our robust PET-only processing, or RPOP processing stream. We used templates for each of the 3 approved radiotracers and ideas that included prototypical negative, positive, and kind of average or intermediate scans. We were able to warp the images into template space. We were able to estimate the resolution and bring them to a lowest common denominator of about a 10 millimeter smoothing kernel. And then we were able to use the cerebellum as our reference region and the neocortex as our target region to generate centiloid values. So the centiloid scale is a scale that can be used to interpret amyloid PET quantification across different radiotracers and processing streams. And it's really anchored at 0 centiloids, which represents a negative or non-elevated amyloid PET in young individuals who are unlikely to have any amyloid in their brain. And the other anchor point is at 100 centiloids, which represents the mean uptake in patients with mild to moderate dementia due to Alzheimer's disease. Values can be lower or higher than these, but these are the anchor points for the scale, just to give you some intuitive interpretation. As I'll discuss, the threshold for a positive scan is usually around 20 to 40 centiloids. In this study, we used 24 centiloids as a threshold. Above that value would be considered a quantitatively positive scan. So this is a very busy slide, but I'll just give you the bottom line, which is that we, in IDEAS, archived images from 10,774 patients, and bringing these images through our in-house quality control and pipeline, we were actually able to quantify results from 10,361 patients. So 96% of the scans, despite some of the real-world adventures that I showed you, were good enough for quantification. These are the demographics of the individuals in the imaging data set that we analyzed. They're very similar to the demographics of people in the overall study. So mean age of 76, about 51% female, about 37% were imaged at the time they had dementia, 63% with MCI. Here you can see the mean MMSE of 24, mean MoCA of 21 in the study. 85.5% self-identified as non-Hispanic white, so there was limited diversity in this data set. Sort of demonstrating the real-world aspects, about 71% had vascular risk factors or known vascular morbidity. And this is the breakdown by the different tracers. So 29% were imaged with florbetaben, 65% with florbetapir, 6% with flutometamol, and overall in the community, 61% of the scans were visually read as positive. These histograms show you the centiloid values that we derived from these images, color-coded by radiotracer. So florbetapir in orange, florbetaben in green, flutometamol in purple. And this histogram shows you that if you have looked at amyloid PET quantification before, you'll know that this is a very typical bimodal distribution, with one peak of negative scans anchored around 0 to 10 centiloids, and a second peak of positive scans anchored somewhere between 60 to 90 centiloids. And this is just visualization of centiloid values with the different tracers, showing you that our pipeline was successful in deriving comparable distributions and comparable looking images for each centiloid value across the different radio tracers. So this is sort of a first pass showing proof of concept that we can actually quantify real-world amyloid PET scans, and the results are very similar to what we've seen in research studies. As I mentioned, we used a threshold of 24.4 centiloids. This was a threshold determined from an independent data set that had autopsy confirmation. So this was a threshold that best discriminated patients who had intermediate or high overall Alzheimer's disease pathology from those who had none to low Alzheimer's pathology. Here's the distribution of centiloids. Now, in the scans that were visually read as positive in orange and visually read as negative in blue, you can see that the positive scans had a median centiloid value of 74, the negative scans a value of about -2. And here we have broken this down also by dementia and MCI. So when scans were read as positive, the centiloid values were very similar, 76, I'm sorry, these are mean values, 76 for dementia. I'm sorry, let me correct myself again. The median overall in the distribution is 57, but the median here in the dementia group is 76. In MCI, it's 73. This really fits with amyloid reaching an early plateau in the disease, even at the MCI stage. So not much change between MCI and dementia. The scans that were read as negative were anchored at around -2, -3 centiloids. So again, just showing plausibility of the quantification. Now here's an important slide. In this slide, we compared our quantitative classification using that threshold of 24 centiloids and the visual reads in the community. And what we found was actually very good agreement. Overall, 86.3% agreement between the local visual reads and the quantification, which translates to a Cohen's kappa value of about 0.71, which is considered very good. 53% of the time, both the visual reads and the quantification were positive. 33% of the time, both were negative. And you can see when the scans were discordant, it was very similar rates, about 8%. The scans were visually read as positive, but quantitatively were negative, and about 6% of the time, vice versa, visually negative, but positive based on our quantitative threshold. Here are the distributions again, so you can see a much sharper bimodal distribution, less of a middle area in the scans that were concordant between visual reads and quantification, and a unimodal distribution in the discordant scans centered right around that threshold of 24 centiloids. Now, looking at the literature, thresholds have been sent anywhere between 10 to 40 centiloids. And so if we were to use a two-threshold approach where we consider everything above 40 centiloids clearly positive, anything below 10 clearly negative, and everything between 10 to 40 centiloids might be considered in an intermediate range. And when we looked at the concordance of visual reads and quantification using those two thresholds, you can see that when the scans were concordant between visual reads and quantification, very few of them were in the intermediate zone. But in the discordant scans, 37% of them were in that intermediate zone, suggesting that the scans were really difficult to interpret both visually and quantitatively. This graph just shows you the range of Cohen's kappa on the y-axis across different thresholds to show you that our good kappa wasn't really dependent on our threshold. So anywhere between 10 to 40 centiloids, we have kappa values exceeding 0.65 between visual reads and quantification. In fact, in a data-driven approach, the optimal threshold for agreement was 24.6 centiloids, so almost identical to our a priori set 24.4. Now, in ideas, radiologists, nuclear medicine physicians were allowed to use quantification if they wanted to, and about 16% of the time they did. When they did that, we got better kappa values around 0.79 and an optimal threshold of 19.7 centiloids compared to those who did not use quantification, a little bit lower kappa and a little higher threshold. So this suggests that if you combine a visual read with quantification, you can get more reliable reads and probably detect amyloid at a little bit of a lower threshold, so higher sensitivity for early amyloid accumulation. So about to wrap up, but here we really compared quantification and visual reads Another way to look at that is to ask, well, how well did the local reads compare to expert reads? And so this is a study that we presented last year at the Clinical Trials in Alzheimer's Disease Conference, where we had 500 scans for each radio tracer selected by our data core. The scans were representative of the overall demographics of the sample, and each manufacturer selected 3 expert readers to overread these scans retrospectively, blinded to clinical data and local reads, and then we asked what was the agreement between the majority expert readers and the local readers. And again, we got very good kappas. Here you see overall a kappa of 0.76 across all the radio tracers, and the values range from 0.72 to 0.78. So very good agreement between local readers and expert readers. And so to just wrap up my portion, IDEAS was the first large study to really assess the performance of amyloid PET in a real world setting. As I showed you, we had some interesting quality for some of the real world data, but we were able to show overall that quantification was feasible in the vast majority of the community acquired scans, and we found very good concordance between local visual reads, expert reads, and centiloid quantification. Our study, I think, also underscores the potential utility of adding quantification to visual reads, which may improve the sensitivity and accuracy of the clinical interpretations. And I think really importantly, if we're going to use PET to follow treatment effects, look at removal of amyloid on PET, that quantification is going to be really critical as opposed to just qualitative visual reads. IDEAS also provided an opportunity to compare emerging blood-based biomarkers from Alzheimer's disease with amyloid PET in a real-world setting. So Dr. Bronstein will talk about that, I'm sure. And importantly, this data set, including the imaging and the clinical data set, are now available for qualified investigators. We're happy to share them through the Global Alzheimer's Association Interactive Network, or GAIN. And so we can provide information about how to apply if you're interested in looking at some of these data ourselves. And so And so I'd like to conclude by just thanking the people who were instrumental in doing this work, including Renaud Lajoie and Ehud Zeltzer at UCSF, Leonardo Iacarino at the time at UCSF, now at Avid Radium Pharmaceuticals, who developed our in-house pipeline. And these are the individuals who helped contribute to our quality control and standardization and quantification of this massive number of amyloid PET scans. And a huge thank you to the IDEAS study chairs, including Maria Carrillo from the Alzheimer's Association, the American College of Radiology, Medicare, and our manufacturers who helped with funding for the study. And thank you very much. And at this point, I will turn things over to Dr. Bronstein for his portion of the webinar.
Great, Gil. That's a tough act to follow, but I will try. Great to be part of this panel. And what I'm going to do is share my screen. So Corey, I'm hearing a bit of an echo. Okay, seems to be a little bit better.
Seems to be okay now, Joel?
Yep.
Perfect.
That's great.
Okay.
So, okay, so hopefully everyone can see my screen. Really, I think we'll just take a little bit of a step back because I think that was a terrific overview, and the way in which policymakers like CMS and academic medicine and advocacy groups and research organizations like Alzheimer's Association and clinicians who are out there taking care of the bulk of the patients can come together to really understand the clinical utility and the importance of these modalities to help improve the diagnostic accuracy for a patient that's experiencing Alzheimer's disease symptoms or other concerns for other cognitive conditions. And so I think that just, again, taking a little bit of a step back, you know, what's the rationale for blood-based testing? This is a schematic that I think shows just the great degree of complexity and challenges that patients face in what we call this diagnostic odyssey. And there's a lot of discussions even among, we've seen even among certain payers that ask the question, like, what's the importance of knowing, and what's the clinical impact of having a more precise diagnosis, and how does it improve clinical outcomes or clinical care processes. So I think that obviously, there's a fundamental basic right for patients to know their healthcare information. And coming from the field of cardiovascular medicine, and certainly having some experience in other areas of internal medicine, generally speaking, patients wind up having the best clinical outcomes when they're more involved with their own care, and also when they and their clinicians are more informed as to what the specific diagnosis is. And I think that obviously, goes beyond just understanding what a person is experiencing at the biological level, so that one can tailor treatment strategies to those patients, at the appropriate time and appropriate clinical circumstance. There's also what's become this vital need for early diagnosis, and the rationale for early diagnosis does relate a bit to the utility of disease-modifying therapies as they are finding their way into routine clinical practice. We're really fortunate to be at a point in Alzheimer's disease care where we have emerging treatment options that are really helping to provide clinical benefits for patients. But what's really important is to recognize that these therapies appear to be most effective when intervening at the earliest stages of the disease process. So that, again, puts a high priority on the healthcare system to ensure that patients are getting a timely assessment. And then, of course, that lost time equals a lost opportunity for intervention. Of course, we're dealing with multiple barriers when patients enter the healthcare system. Obviously, there's limitations in the diagnostic yield from the history, physical exam, and basic imaging assessments. There's access challenges and healthcare disparities. Obviously, amyloid PET represents an extremely accurate modality. While it does have some limitations, it does represent an accurate strategy for identifying pathology, but it's also associated with high costs. It does carry a certain amount of radiation exposure, and it also has differential access for many different patients based on where patients reside, whether they're in rural areas or other underserved communities. They may not have access to these facilities to be able to get such high quality testing. And of course, lumbar punctures and cerebral spinal fluid analysis are also accurate modalities, but they have a perceived high level of invasiveness by many patients. And in about 15 to 20% of patients, they will be contraindicated to receive such therapy because, or such assessments because of bleeding disorders or other contraindications. And so what we've seen with these significant barriers to access and the increasing accuracy that we're observing with blood biomarkers, there's a growing consensus among the clinical community, KOLs, policymakers who are thinking about how to evaluate the incorporation of these blood tests into clinical care to now really support the use of these blood biomarkers. So when we talk about blood biomarkers, we're going to, for the most part, talk about amyloid pathology today, but I think another very important part of fluid biomarkers and even imaging is the ability to look at tau pathology. And when we think about the pathological spectrum of disease, patients usually display amyloid pathology earliest, with tau pathology emerging later. And tau pathology really correlates strongly with clinical progression. You tend to see amyloid pathology occurring in the earlier stages, many times when patients are asymptomatic. And as patients become symptomatic, you begin to see the earliest manifestations of tau pathology. And so there's clearly a good use case for both markers that measure amyloid pathology and tau pathology. Now, this phosphorylated tau 217, which we'll talk quite a bit more about in some subsequent slides, is actually a really good marker to picking up early amyloid pathology, and it does have some nice correlations with early tau deposition. But if we really want to look at the earliest stages of amyloid deposition, that's where A beta 4240 as a fluid biomarker continues to contribute in a meaningful way to be able to identify some of those early abnormalities. And then when we start thinking about more advanced stages of tau pathology, like Braak stage 3, 4, that's when you start getting into the clinical progression. Biomarkers like p-tau205 or MTBR tau243, which is a direct measure of tau tangles, can play an important role. And C2N is actually developing all of these biomarkers. We'll talk about the Precivity-AD2 test, which is available for clinical care today. I think another important point when we talk about blood biomarkers in clinical practice is to recognize that there's many different potential use cases. And really the best studied clinical application today and where the clinical need would be most significant is to identify, help identify amyloid pathology in a person who's experiencing early signs or symptoms. And again, part of that driver is coming from the fact that we now have disease-modifying therapies that can help address early stage Alzheimer's disease. These people have early stage symptoms, usually at the MCI stage, to receive such therapies. And so these patients who present to the clinic with a cognitive concern are really looking for methods for proper differential diagnosis, and blood tests are emerging as a vitally important tool for that. But of course, there's future applications whereby blood biomarkers are likely to play a very important role. We're not going to spend a lot of time talking about these, but these are areas of very active research. There's some very promising early data to suggest that, in fact, we will be able to see maturation of these fluid biomarkers for helping in risk assessment, which would be to identify people who are actually asymptomatic and at high risk for developing future clinical disease, looking at monitoring, being able to assess changes of disease severity or the effect of a treatment over time, and then using these markers as prognostic markers that can help identify a baseline assessment of one of these tests to predict how well a patient may respond to a treatment. And because these treatments are all associated with certain benefits and costs, that risk-benefit analysis is really important, and a prognostic biomarker could be very useful to help identify the most likely candidates of patients who are going to derive clinical benefit. So when we talk about some of the regulations, we're not going to get into a lot of detail here, but C2N Diagnostics operates its clinical operations under CLIA rules, which is administered by CMS, and a CAP accreditation, which is the College of American Pathologists. And that really speaks to the quality controls for a laboratory. These are rigidly monitored requirements of clinical labs. And I think that this CAP accreditation in particular represents kind of the highest quality of achievement for a clinical laboratory. And it's so important for having trust in that laboratory, because it's the laboratory that's providing these diagnostic tests to provide interpretation and results that are going to ultimately come back to patients. Now, the CAP CLIA rules are not mutually exclusive with FDA, and some of you may be aware of some of the evolving regulations where the FDA is trying to impart more control and regulation around these laboratory developed tests, which are tests that are brought up within a single lab. And C2N fully respects that process. In fact, C2N does maintain what we call a breakthrough device designation with the US FDA, which acknowledges that it's a test that represents a novel offering and in fact can address an area of significant unmet medical need. And so this is an area of ongoing effort within the C2N lab, where we are developing will ultimately be cleared by the FDA. And oftentimes these tests are virtually identical to the tests that's being offered under CAP CLIA. And so C2N has a lot of experience offering these high quality tests under a regulated system. And so when you look at some of the clinical care test components, C2N uses mass spectrometry. We'll talk a little bit about the virtues of mass spectrometry, why we use that as an analytical platform. But this is a schematic of what it is we actually measure. And the Passivity82 test in particular has been a breakthrough product because it does a really nice job of correlating with some of the reference standards that we're used to seeing. For example, the PET data that you just saw from Gil and CSF measurements. And so it's the quest towards getting the most precise and accurate blood tests that are really going to move the needle in clinical care. And the Passivity82 test really represents a breakthrough. But we were really fortunate to be able to work with the IDEAS cohort, which you heard earlier in the presentation, to try to validate these blood biomarkers in a real-world clinical setting. So we actually approached Alzheimer's Association and Gil and the rest of the leadership committee at the IDEAS study, and we said back in 2018, 2019 timeframe, we said, hey, we're developing these blood tests, and we think these blood tests could be super important in clinical care, and we'd love to be able to bring patients who participated in IDEAS back to get a blood draw and be able to perform correlation analysis of our blood test with the real-world patient experience from IDEAS. And that's exactly what happened. That became what was known as the PARIS sub-study. And the PARIS sub-study became one of two pivotal clinical trial data sets which were used to validate not only Passivity82, but the Passivity-AD test. So the IDEAS, which started out as a real-world clinical usefulness study that Medicare tried to deploy to understand PET utilization, really was able to receive significant leverage by our introduction of the PARIS study and using that data set to help validate what really became the first two clinical blood tests for aiding in the diagnosis of Alzheimer's disease. So, there may be some questions out there as to why C2N Diagnostics is so passionate about mass spectrometry and what it is about the platform that we really like compared to, let's say, immunoassay platforms. C2N doesn't necessarily have any kind of restrictions to be working with mass spectrometry for commercial reasons, but we like the analytical precision and the power of mass spectrometry. So mass spectrometry is really, in the most simplistic form, a very sophisticated scale for looking at subatomic weight differences and ionic charges of biomolecules. So when you take proteins that have extensive post-translational modifications, for instance, a phosphorylated tau protein fragment, one can actually use this mass spectrometer to precisely identify these fragments with an unparalleled degree of sensitivity and precision and accuracy. Mass spectrometry represents the gold standard for protein quantitation in other biomolecules. And the reason why is because every molecule, biomolecule, has a unique ionic charge and subatomic mass that, with proper resolution of a mass spectrometer, can help separate one particle from another. So we try to use this underlying kind of physical properties of mass spectrometry to understand biology of diseases like Alzheimer's. And so in particular, we know that, let's say, this phosphorylated tau 217 fragment is really important with regard to its correlation with amyloid pathology. This hyperphosphorylation of the tau protein does appear to be a core component of Alzheimer's disease pathology progression. And so we're interested in obviously measuring the p-tau 217 fragment. However, we're also interested in controlling for individual level variability that can occur for any analyte that's being measured. And these systemic factors like body mass index, overall circulating volume, as in patients with impaired ejection fraction, patients with cerebrovascular disease, people who have kidney impairment, who may not metabolize these peptide fragments properly compared to normal controls, can lead to a lot of perturbations in these analyte measurements. And so the question becomes, when you identify an individual with an abnormal p-tau217 value, is it due to the underlying pathology in the brain, or might it be due to some systemic confounder that could be artificially elevating that value? And so what's really nice is we have the ability to multiplex with mass spectrometry. And so we can take a marker like NP-tau, which is the non-phosphorylated tau217 fragment, and we know that that marker has no correlation with brain pathology. And then what we can do is we can actually express the p-tau217 value as a ratio that's been normalized at the individual level based on a person's NP-tau217 value. And that's what produces the percent p-tau217. We do the exact same thing with A-beta-42, which tends to be the analyte that's more correlated with amyloid pathology. A-beta-40 serves as that reference standard peptide that we can use to help normalize against potential clinical confounders. And so we can combine these multiplexed analyses with the mass spectrometer output into what we call an algorithm. The algorithm is calculating the amyloid probability score, which we reviewed a little bit earlier, and we'll talk a little bit more about. This APS is an indication of the likelihood that a person has amyloid pathology in the brain. And so we'll talk a little bit about the two studies that were used to clinically validate both PrecivityAD and PrecivityAD2. And the first study came from the Paris cohort. And the Paris study, we actually divided into two cohorts. One was what we call the discovery cohort. The second is the validation cohort. Now, it turns out the validation cohort we will be reading out very soon. It has never been read out, and it will be used for supporting our FDA registration. So the Paris discovery cohort, which was a segment of the total Paris cohort, about 250 some odd patients, were combined with the Mission AD study, which was a phase 3 clinical study in MCI and early AD. This was a program ran by AZI. They generously enabled us to use the baseline samples, and we were able to look at the performance of initially the PrecivityAD test, and then ultimately the PrecivityAD2 test once it was developed. And every one of these patients had an amyloid PET scan within 18 months of blood sample collection. Now, we talked a little bit about Centiloid in the earlier part of the presentation with Gil's overview of ideas, and we actually used the Centiloid score of 25, which at the time we didn't know, but has subsequently been shown to have a very strong correlation with the optimal separation of patients who are amyloid positive and amyloid negative by way of neuropathology. And so we used, which was 24.4. And so we used the Centiloid score of 25 to identify patients as amyloid positive or negative. And so the PrecivityAD test we've already reviewed. I'm not going to go into a lot of details, but we've extensively validated that. The first paper was published back in 2022 with the assessment of the APS to evaluate amyloid PET findings among patients with cognitive impairment. That was published in JAMA Network Open. We subsequently went in to validate the PrecivityAD test using pre-specified cutoffs, the algorithm, and tried to say, well, how well does it predict disease in an orthogonal clinical data set like the ABLE cohort. And so we chose patients from ABLE who had MCI and early dementia, and were able to demonstrate that, in fact, the PrecivityAD test had high degree of robustness. So when you look at these two clinical cohorts that were used for both PrecivityAD and AD2, you can see that in general, the patients were average age of 73, 49% female gender. We had 30% Hispanic population. Many of these patients came from the Mission AD study. We had approximately a 80% presence of MCI versus 20% dementia. And you can see the distribution of APOE4 carrier status among people who were Centiloid negative and Centiloid positive. And as you would expect, patients who were amyloid positive had quite a bit higher ApoE carrier status. And then you can see that the overall population had a 52% prevalence of amyloid PET positivity based on visual read, which was highly concordant with the centiloid measures, where we saw 90% of those patients who are visual read positive also being centiloid greater than 25. And so the Percivity-AD2 test we introduced into the clinical market about a year ago, and again, using the Mission AD and PAIR studies to help validate it. And we've introduced this test for use in patients 55 and older with signs or symptoms of MCI who are undergoing evaluation for AD or other causes of cognitive decline. And what's important about the APS2, which is again this algorithm that includes ApoE A-beta 42/40 ratio plus p-tau217 ratio to provide this value between 0 and 100, we use a single cutoff value to negative or positive in the interpretation of having amyloid pathology consistent with a neuropathological diagnosis of Alzheimer's disease or absence of amyloid pathology. I think another really critical point about this blood test and really any modality that's used to help in pathology assessment is that these are really only clinical tools to be used in the context of a physician's and healthcare provider's judgment. And it's really the totality of the evidence in the patient's clinical presentation that ultimately renders that clinical diagnosis of Alzheimer's disease. But these pathology assessment tools wind up being extremely important to improve diagnostic accuracy. And what you can see here is actually a histogram that separates the patients based on the APS2 value from 0 to 100 on the vertical axis, and separating patients by amyloid negativity with the centiloid less than 25 or the centiloid greater than 25. And as you can see from this histogram, the distribution plot actually looks very similar to what we saw with the amyloid PET scans. And when you look at the APS2 test being compared to amyloid PET as the reference standard, the test achieved an overall accuracy of 88% with sensitivity of 88%, specificity of 89%, and among patients with a 53% disease prevalence of amyloid positivity, this correlated with a PPV of 90% and NPV of 87%. So this level of performance is very comparable to what is seen with cerebral spinal fluid analysis. We'll talk a little bit more about some of the data that we've recently generated comparing the test against CSF measurements. And so what we found with the APS2 is that it showed more robust performance versus the individual biomarker measurements or the biomarker ratios. The APS2 was more robust than the percent p-tau217 in this study, which was more robust than the p-tau217. People always ask the question, well, what's the key difference between the percent p-tau217 and the p-tau217? We talked a little bit about that earlier, but the ability to incorporate these analyte ratios allows us to mitigate differences in race and comorbidities that can confound the results of a straight p-tau217 measurement. And ultimately, this APS2 measure was the most robust metric to distinguish patients for amyloid positivity or negativity. And then I think it's also really interesting to be able to think about the clinical context by which a blood test might be used. In the circumstance where you have a patient who comes into the clinic like with a different risk profile, whether that patient represents a person with like symptomatic complaints but no clear evidence of signs of disease, you might put that person into the subjective cognitive decline population, where we know the baseline prevalence of amyloid pathology is close to 30%. You can see that the PPV and the NPV of the test is very much a function of disease prevalence. In this case, your NPV for the APS2 result at the cutoff value that we've chosen would yield an NPV of 95%. So for that SCD population, when you have a negative test, you can be very confident that in fact there is no disease. Likewise, when you have a person who may fall into a high-risk category like dementia, is a natural property of diagnostic tests. When your disease prevalence increases, your PPV will increase, your NPV will decrease. So in that patient who has clear signs of cognitive impairment and maybe at the early stages of dementia, that would correlate with a PPV of 94%. The other thing we can do is we can look at the performance of the APS2 across different subgroups. And so in this study, we looked at how it performed in the Paris data set, the Mission AD data set. We looked at how it performed comparing whites versus non-white individuals. We looked at the robustness in Hispanic population, different age groups, and ApoE4 carrier status. We've actually subsequently gone on to look at different comorbidities, and the test winds up being extremely robust regardless of the potential clinical confounders. The other point to kind of emphasize here with regard to the PPV and NPV is that because the APS2 is set with a floor of 0 and a ceiling of 100, the APS2 result itself winds up being quite useful. So if a patient, for instance, has a score of 6, you can have a very high degree of confidence that the patient has no pathology. And likewise, when a patient has a high APS2 like 93, you can be certain that that person has a very high likelihood of amyloid pathology. And again, this is based on the way we constructed the algorithm, such that at lower levels, it's equating to a higher degree of certainty that there's no amyloid pathology, and at higher levels of the APS2 associated with more certainty. So I only have a couple of more slides, and I think many of you may have already seen some of this data, but we were really privileged to be part of an amazing collaboration with Oscar Hansen and Sebastian Palmquist and a bunch of colleagues in Sweden to look at how the APS2 performed in both primary care and secondary care, and importantly, how the test performed using a pre-specified cutoff value, because we've been using this test clinically for over a year based on the original validation data. And it's always important to be able to validate in orthogonal data sets and even prospectively to ensure that that predefined cutoff value is providing the kind of diagnostic robustness that you would want in clinical care. And so what was interesting about this population was over 1,200 patients with a very high degree of comorbidity. You can see that 70% of patients in primary care had cardiovascular disease. That actually matches up quite well with the patients who presented to the IDEAS cohort with about 71% frequency of cardiovascular comorbidity. Kidney disease was prevalent in about 26% of patients who presented to primary care, and even 17% among secondary care. And so I think this slide really demonstrates, one, that you do get a broad display of patient clinical presentations with regard to stage of disease, where you see people with SCD, MCI, and dementia, and then patients don't come in with a simple presentation. They often have frequent comorbidities that can complicate the interpretation of the test result. And so what we found when we looked at the data using pre-specified cutoffs and prospective testing, where we receive samples on a weekly, bi-weekly basis to look at the performance, you can see that the overall accuracy of the APS2 result was over 90%. And what was really interesting is that when you look at the clinical diagnostic accuracy among the dementia specialists, they had a clinical diagnostic accuracy of 73% using standard of care evaluation tools. And primary care, who again represent really the primary gatekeepers to seeing these patients with cognitive concerns, they only had an accuracy of around 63%. And using the blood test, one was able to achieve over 90% accuracy for both primary care and specialist care, which really shows the overall robustness of the test. And importantly, 80% of the patients in this BioFinder study were actually evaluated using CSF as the reference standard. And so the key findings from this study, and really consistent with all the data that we've evaluated from many different studies at this point, showed very high concordance of CSF analysis in distinguishing patients with and without AD pathology. AUCs 0.96 to 0.97, which is really about the best you can expect when you're comparing to an imperfect reference standard. Consistent and robust performance in prospectively collected samples, demonstrating clinical accuracy that was superior to the current standard of care evaluation tools. And then importantly, the performance continued to stay very high regardless of comorbidities and the stage of clinical disease. So I'm going to stop there. C2N is extremely committed to evidence generation. We're happy to provide you with access to the many publications that we've been part of. We've been involved with both the donanemab and lecanemab studies to help demonstrate biological activity. Over 150 clinical studies throughout the world and over 30,000 precision-related biomarker measures have been reported in publications, with many more to follow. And so, we'd be delighted to be able to share those with you upon request. So thank you.
Thank you so much. Thank you so much. And there's quite a lot of questions that have come in. So in the interest of time, maybe what I'll do is just ask, a quick question to each one of you. So it sounds as if blood biomarkers are definitely ready for clinical practice. Compared to PET, and where the RAND report just came out that PET is not readily available, how would you say that, talk about the information that HCPs and patients may need when they're deciding which Alzheimer's disease biomarker to rely on?
Yeah, it's a great question. First of all, it's exciting that we have access to these different biomarkers. I think one thing we haven't kind of talked about is cost and coverage, and that's definitely a consideration when thinking about biomarkers. I think that the blood biomarkers, especially the high-performing ones, are doing so well. It's likely that they're going to replace the need for lumbar puncture, at least for measuring amyloid beta and phospho-tau. There are other things you might get in an LP if you're worried about inflammation, et cetera. But just for those biomarkers, I think the high-performing blood biomarkers are performing similarly or equivalent to CSF. PET is measuring something different. So it's not measuring, concentrations of monomers, but it's measuring the overall pathology in the brain. And there may be use cases where that is critical for providers to know. For example, if you're starting someone on a therapy, you might want to know the centiloid value at which they're starting, and then if you're imaging them again in a year and a half, how much has that dropped, and can you pause or modify the treatment? So there are different use cases, I think, for the different biomarkers. I anticipate there will be some clinical practice guidelines that will come down the port soon that will help advise clinicians about which biomarkers are suited for which use cases. But overall, it's just exciting that we have access to this. It wasn't too long ago where it was just our clinical diagnosis, which, as Joel showed, is even for experts is not ideal. And now we really can have very accurate tests for what's going on in the brain at the molecular level.
Great. Thank you so much, Joel. Any wrap-up thoughts? And we do have our contact details. Please send us an email should you have any questions. But Joel, I'll let you finish up and say thank you.
I would just like to say, I know it's all my fault. We didn't leave a lot of time for questions. If people have questions, please submit them. We're happy to address them, and we'll even bring Gil into the exercise to make sure they're properly addressed. It's really important to be able to address all your questions.
Okay, great, excellent. Thank you, Joel. And Sheena, I'll just jump in here to wrap things up. As you were saying, unfortunately, we have run out of time, but we did have a lot of great questions come in. And as Joel was just saying, if you have any questions, any further questions, or if you're someone who's watching the recording of this webinar, you can absolutely reach out to the team. We've got a couple email addresses up there on screen for you. And as Joel mentioned, C2 would be very happy to answer any and all questions that you have. We do thank everyone once again for participating today. I have added a link in the chat box where you'll be able to view the recording of of this event, and you can, of course, share this link with your colleagues so that they may register to view it as well. You will also receive a follow-up email from Xtalks, and after we wrap things up here, a survey window will appear on your screen. Participation in the survey is always appreciated, as it does help us to improve our webinars. Please join me in thanking our speakers once more for their excellent presentation. Sheena, thank you for being here. Gil, Joel, thank you so much for the presentation. We hope our audience found this informative, and we wish you all a great rest of your day. Take care, everyone.